Thanks everyone for joining the fifth season of Crops TV. We're so glad that you subscribed. Today I get to talk about my favorite subject, which is soybean aphid, because it was such an important pest in 2024. So, along with Ashley Dean and myself, I'm Erin Hodson. We've prepared some field notes, some extra resources for you with the highlights and places to get more information. And we'll get started talking about soybean aphid. I hope by the end of our time together, you'll have a better understanding of the seasonal dynamics of soybean aphids. For some of you, this may be totally new. For others, hopefully, it's just a refresher. I do want to talk about some of the resistance issues we're having with an insecticide and provide some alternative management tactics for you for future growing seasons. Aphids have complex life cycles, which is interesting to me. Most of the aphids, which are considered field crop pests, have a primary and a secondary host. In the case of soybean aphid, their primary host is buckthorn, which is a woody plant found in the understory of shelter belts and forested areas. And their secondary host would be soybean. So they alternate between buckthorn and soybean every year. They do overwinter as an egg on buckthorn, and with accumulating degree days in the spring, they hatch. They'll have a few generations on buckthorn before they move over to soybean, in which they can have 15 to 18 overlapping generations. Now, it's important to note that in the spring and the summer, they are having asexual clonal reproduction. So it's females giving birth to their genetic clones. And it's not until the environmental cues tell them to move back to their primary host, such as shortened photoperiod, cooler temperatures, and declining host plant quality of soybean, that they make that fall flight back to buckthorn, in which males arrive, they'll mate, and produce an overwintering cold hardy egg. So, this is a complex life cycle, but one that has proven to be very successful for field crops because of the number of generations they can produce in a single summer. So, in addition to the clonal reproduction, there's a few other ways that they can really speed up their life cycle. And the first one is that they give live birth. Unlike most insects that lay eggs that take some time to develop, aphids are born live. You can see a picture here. In fact, side note, they're all born breech. And that way, the nymphs are ready to feed that same day and can greatly speed up the life cycle. In addition, they have telescoping generations. And I'm stealing a quote from someone that said, aphids are kind of like living inside of your grandmother. So, this is an older drawing that shows basically aphids, their abdomens are just two big ovaries. And the nymphs or the daughters that are developing inside the adult are also developing their own daughters at the same time. So, this is the telescoping effect that aphids can have. So, you have an adult female that has daughters and granddaughters inside of her at the same time. So this is a picture of soybean aphid, and you can actually see the eye spots of the nymphs that are about to be born. I was part of some research for soybean aphid when it first came to North America in the early 2000s, in which we were trying to uh, describe the optimal temperature for soybean aphid. So when do they do their best? And it's right around 77 degrees. So of course, this is in a lab, a very controlled situation. But when it's 77 degrees, they can live up to 22 days. They can produce up to 75 nymphs, and you can have this exponential doubling time of 1.5 to 2 days, due in part to that they give live birth and their genetic clones and that telescopic generation. So you could see how you could have a few aphids in the fields, and if you had 77 degrees for about a week, where you have the exponential growth, you come back and there's hundreds of aphids per plant, which has happened several times. So, what's normal or what's healthy for soybean aphid? You can see a picture here of an adult aphid surrounded by lots of nymphs. They're on the small side, 1.5 millimeters in length. They have bright limey green bodies with red eyes. All aphids have cornicles or tailpipes, and in the case of soybean aphid, they're dark. This is different than all other insects that don't have cornicles. And in general, they're pear-shaped body, and a nice, healthy soybean aphid is kind of juicy or plump-like body. Now, they may be confused with potato leafhopper nymphs because they're relatively the same size and color. But if you look at the picture on the bottom, the nymph has a big wedge like head, large white eyes on the side of the head, and their abdomen tapers. So they're kind of built opposite than a pear shaped aphid. And in addition, their behavior is quite different. Soybean aphids are kind of lazy or sedentary, and once they tap into the plant, to take up phloem, they're likely not to move very far in their life. And this is compared to potato leafhoppers that are very skittish, very active. They're going to fly, jump, or walk away from you if they're startled. Now, most of the time, you're going to see aphids in the wingless form, but there are times throughout the spring, summer, and fall in which they can generate a winged form. So, an adult will eventually tell the granddaughter inside of her that it's time to produce wings. Again, from environmental cues, looking for food. Maybe it's too crowded. Maybe the photo period is changing, or maybe there's just a lot of predators around and it sends out an alarm signal to the colony. So this is a soybean aphid on the bottom. She has nice, big, clear wings that kind of act like sails. She's not a great flyer on her own. She can move within fields or between fields, but for long distance movement, she needs to go up into the atmosphere and catch those jet streams to move, say, from Minnesota to Iowa. So notice the darkened thorax. She has a lot of muscles in her thorax to help her pump the wings. But in any situation, you're likely to see mostly wingless, but sometimes winged individuals mixed in together. What's not normal or what's not healthy for soybean aphid is to see them various colors. They could be white, brown, black, or even orange. And their shape changes as well. They can become flattened or just really small. Even the adults can be under a millimeter in size. Sometimes they could look fluffy or fuzzy or even powdery, kind of like a little powdered donut. So, in these situations, you could have an entomopathogenic outbreak or a fungal outbreak that's naturally occurring in the soil. And like all fungal outbreaks, you have to have the right environmental conditions, such as temperature and humidity. But to see these powdery looking donuts is a good thing, meaning these fungi just wipe out aphids on their own. Also, aphids have a hard time reaching that optimal growth because they're kind of wimpy and they're not very competitive. If there's any other challenges with pests like insects, maybe spider mites, fungal pathogens like brown stem rot, or soybean cyst nematode, they are not going to achieve their ultimate or their optimal reproduction. They don't like it too hot, so if you have an exceptionally long stretch of 90-degree plus days, they're going to not produce as many nymphs as they normally would. And that's when, say, we might have spider mites starting to take over. They don't do well in periods of heavy rainfall. And especially when plants are small, they can be washed off the plant and have a hard time getting back on the plant. And just in general, they don't do as well in the high potassium soils. Predation is also really common for aphids. That's because they're kind of easy prey, especially soybean aphids, like I said, are fairly sedentary and they may not be able to get away from predators like beetles, spiders, flies, bugs, or lacewings. Really, their only defense mechanism is to try and kick their legs, and that's usually not enough to fend off some of these very voracious predators. So it's a good sign to see these in the field, although it's hard to have a good community of predators if that field happens to be on a frequent pesticide exposure because almost all of these are going to be highly susceptible to broad spectrum insecticides. The same goes for parasitic wasps. We have a couple of species in Iowa that specifically attack soybean aphid. There's a picture on the left that shows an adult wasp that is queuing in on that soybean aphid. She is going to inject an egg inside the abdomen, and that wasp egg is going to hatch, turn into a larva. That larva is going to consume the contents of the inside of the aphid, eventually killing it. At this point, they start to become bloated and off color, and they could be brown or tan or black in color and would be referred to as a mummy. And they often look glued or kind of stationed on a plant, and that's just because they're not alive anymore. When those wasps complete development, they're going to create a little escape hatch. And so it's not common to see the wasps, but it is fairly common to see mummies in the field. Okay, that was a refresher on biology and dynamics. I did want to spend some time reviewing integrated pest management or IPM to protect yield. And this is especially important and useful when it comes to soybean aphid. I've adapted a graphic from two emeritus professors at Iowa State University, Pedigo and Rice, who literally wrote the book on economic entomology that's used in classrooms. And what they said are there are a lot of different components that could be used in a successful IPM program. So, depending on the pest, depending on the system or the crop. The components on the bottom are generally things like myself, entomologists try and understand through research the life cycle, population dynamics, how to properly sample for it, and just trying to understand the behavior of the pest so that we can try and interfere with the success by developing strategies that you see kind of at the top, using pesticides, natural enemies, host plant resistance, and even cultural control in some cases. And what we want to do again is use multiple tactics, hopefully in a proactive manner, so that we're not always using rescue strategies like foliar insecticides as a last resort. So if you notice at the keystone of it all is economics and applied entomologists like myself are trying to better understand all of the input costs of that system or that crop and the control costs that might be happening for that pest and also it's harder to predict but also the market value that we expect to get for that crop. And so having a better understanding of the economics or things that we think about when we develop thresholds and is really the keystone of IPM. And so one of the things I helped out with when soybean aphids came to North America is understanding the economic injury level or the lowest point at which will cause economic damage for that crop. So fairly easy thing to do in field crops. How many insects does it take before we can measure bushels per acre being removed for that crop? And for soybean aphid, it has been determined through a wide range of geographies and soybean genetics. It's around 675 aphids per plant is a point at which we can start measuring economic injury. And this is a point at which we never want to get to, but it's not so much the 675 because soybean aphid could be feeding for 60, 90 plus days. What I've done and others have done when we are screening efficacy evaluations is to convert those weekly counts into the seasonal exposure or cumulative aphid days. And so, a really important benchmark that I use and others use as well is 5,500 cumulative aphid days, is the seasonal exposure we could expect to start measuring economic injury. Now, again, a point at which I never want a field to get to. And so, we also establish an economic threshold or a point at which we want to take action with a rescue treatment. It's usually expressed as pest density or plant injury. And for soybean aphid, we would say the threshold is 250 per plant. That is a point in which most situations, if you're making a timely treatment, you're going to avoid the economic injury level and you're going to cover your control costs and hopefully make a profitable decision that day. I personally think pest management in the north central region of the US is a roller coaster. And that's just because in part, especially for soybean, we have variable pest pressure and soybean aphid falls into that in which it's not a every year, every field kind of problem like it is for some pests in other parts of the world. It's a sometimes problem. And in addition to that, we have generally increasing input costs. I have a figure from the USDA showing seed costs, the trend going over from 1997 to 2022. At the same time, we have yields shown in the white squares the yield that we can expect to get for those crops. And ideally, you'd like to see those seed costs, that increasing trend, raising at the same rate as yield. But there's a lot of reasons why the yield is more fluctuating, and not only the market values for exports, but things like the environment can decrease the optimal yield that we would expect to get for that crop. And so we have to think about that. There's ups and downs, there's good years and bad years for pest management. In addition to that, we have to think about access to effective pesticides. The products that we use in field crops have to be reasonably priced given the market value that we can expect for the crop. New products are coming on the market, which are generally more expensive. Older products are getting taken off the market. And so we have to think about the access, and also the effectiveness is not always the same from product to product. In the world, we have a lot of invasive species moving all over, and that happens in soybean quite a bit. And so we have new pests coming in, sometimes interacting at the same time as our established pests, and it makes management decisions more complicated. And then, just in general, the world is getting warmer. And with a warmer world, that means insects tend to do better. And same thing with weeds and plant pathogens as well. And so it might be a situation where we have to think about more persistent pest activity with a climate change in the future. And so, if I could simplify my discussion on economics when it comes to especially soybean and soybean aphid, is that farmers have a certain amount of fixed costs to get a crop into the ground. And they also have quite a few variable costs. And I would lump the pesticides into those variable costs. So, it kind of depends on when you buy, how much you buy, the number of times that you're applying, and how you're making those applications, whether you're doing it yourself or you're using a co-op, and fuel, people power, all those things come into play with variable costs. And there is sort of this break even point between profit and loss when we think about revenues and expenses. And so, in my opinion, I think especially there is a chance for us to reduce the amount of insecticides we're using in soybean, because they're not needed and we're not breaking even from some of those treatments. And so, when we think about increasing variable costs, the odds of making a profit become slimmer. So, I'm advocating reducing the amount of insecticides we're using, especially for a variable pest like soybean aphid, because we're not making a return on investment from that application. So I've been looking at soybean aphid populations for a long time, and to say that they have been erratic, they've always been erratic, and I would expect them to be erratic in the future is probably an understatement. I've included just the untreated control plots here over a 20-year period, and also included the economic threshold and economic injury level for your reference. And you can see over a period of 20 years, about half the time my untreated plots will exceed that economic threshold of 250. But only 1 in 4, or about 24% of the time, would they have that optimal growth and exceed the economic injury level. And so this is, again, a sometimes pest. When they exceed the economic threshold, it does not guarantee that they will continue to climb and that field experience economic injury. So they tend to be a very erratic pest within fields and even between fields in a single growing season. So, as I said, I've had efficacy evaluations for a long time, and I see three general trends when it comes to performance. So, this is an example from 2011 in which we have a number of treatments. I took out the treatment names because they don't really matter, but several of those treatments have exceeded a really important benchmark of 5,500 cumulative aphid days. Some are below that benchmark. When we have this trend in which you have very high numbers, some of those treatments are reaching 20,000 cumulative aphid days, we can expect a 5 to 10 bushel per acre yield loss when aphids accumulate over that 5,500. And you can see some treatments on the bottom had very few numbers of aphids, and they're likely reaching that full yield potential. And so this is a trend that I see very often. When I stick to that threshold of 250, I can expect to break even on my control costs and probably or very likely make a profitable decision from that amount of pest activity. On the other hand, about half the time, I don't see enough aphids to reach that benchmark. This is an example from 2016 where the top treatments only had around 1,500 cumulative aphid days. This is the yield response that goes along with those treatments. And you can see maybe 2 to 4 bushels per acre difference between, say, the untreated control and a kitchen sink approach, which might be a seed treatment plus a foliar insecticide. And so this is a point at which I'm likely not breaking even from that treatment. And certainly, I wouldn't expect to make a profit from treating when aphids haven't reached that important benchmark of exposure for the entire season. And then 3 times I have seen this pattern in which aphids came into those plots very, very late in the season. So it was the last part of August or early part of September. I think this example is from 2019. So you can see about half of the treatments did exceed that economic entry level. But the activity wasn't enough to impact yield that late in the season. And so again, it's very hard to tell the difference because there's so much overlap in the mean separation between the treatments. And so when someone asks me, when is it too late to treat for soybean aphid? I generally say R5.5. So seeds are expanding in the pod, but they haven't fully expanded to full seed set or R6. And that's just because I have not been able to demonstrate a consistent yield response when treating so late in the season. So even though there's aphids there, they're not impacting yield like they would earlier in the season. And so if this is my primary pest, I would say it's not worth the treatment time and the treatment costs. However, if you happen to have other pests in the field that are causing direct injury to the seeds or the pods, like bean leaf beetle or grasshopper, that's a different conversation. And I talked about direct injury to soybean in that Crops TV episode last year. So, in addition to IPM, I also like to have people think about insect resistance management or IRM. If this is a new concept for you, there's a really great video on the IRAC website, Insect Resistance Action Committee. And it's a nice animation that goes through how insects develop resistance and some tactics to mitigate that in the population. And the takeaways are basically, as I said, use an effective ingredient. Use high dose active ingredients so that if you're making an application, you want to be using the fully labeled rate and you're not wanting to use sub like half or quarter rates, say if you're using it with a fungicide or herbicide tank mix, just in case type treatment to clean up insects that might be in the field, because it does give a chance for those individuals to overcome that with a less than high dose treatment. For some crops, you might have alternate modes of action. Say for corn, you have different types of Bt proteins. In alfalfa, you have host plant resistance. And for most of the crops, you would have a variety of insecticide group numbers to choose from. So, my recommendation is to alternate with all the tools that you have in your toolbox as long as you think that they are effective tools. And we want to be rotating them instead of layering them because we have relatively few options, especially when it comes to field crops. I recommend limiting the exposure to any strategy that you're using to suppress pests because, again, it provides an opportunity for individuals in that population to overcome. And that's going to speed up or accelerate the rate of resistance in that pest population. And then finally, IRM advocates the use of a refuge, which is actually mandatory for transgenic crops, but highly encouraged with other strategies. We want an area where susceptible individuals can be produced and hopefully diluting those resistant genetics in the pest population. So IRAC provides a nice description of all the possible group numbers for insecticides. Fungicides has a similar website called FRAC, and herbicides does as well, called HRAC. And I've color coded them by how the insecticides kill the insects. And so everything in blue targets nerve and muscles. Everything in green targets growth and development, and it interferes with hormones, it interferes with the hardening of their exoskeleton. And if insects can't become an adult, it really limits the opportunity to generate another generation. All the colors or all the products that are in orange interfere with respiration. And if you can't breathe, you don't live very long. And then the midgut or the brown is indicated by group number 11, which you may be familiar with for BT. This is a microbial disruptor in which the BT proteins attach to the midgut of the insect and cause pores or openings. And so the midgut contents basically cause sepsis to the rest of the body. And if your midgut explodes again, you're not going to live very long. And then there's just a couple of insecticides where they don't know how they work, so they're unknown. But just to say it in another way, most of the products that we're using in field crops are older chemistries, and they do attack the nerve and muscle. So most of what we're using are the products or the group numbers that are in blue. I would say for soybean in Iowa and the vast majority of the north central region, probably 98% of what we're using would be 1, 3, and 4. So they're the organophosphates, pyrethroids, and the neonicotinoids. We also have group 9 and group 28 as options. They're newer chemistries, they're more targeted, and as you can imagine, they're a bit more expensive than the generic older chemistries. So if we take a deeper dive into the group numbers, some of those have subgroups as well. If you pay attention to the salmon colored where it says 3A, those are the pyrethroids. There's a couple different options as far as different active ingredients. And then if you look at the gray, it's pre-formulated mixtures that all include a pyrethroid. So if you look at the options that are labeled for soybean aphid, about half of them have a pyrethroid. And so if you were to take that away because they become pyrethroid resistant, you have some limited options on what you can use as effective choices. So, if you aren't familiar, I just wanted to briefly recap how pyrethroids work. And again, they're targeting the nervous and muscle systems. More specifically, they are attacking the voltage gated sodium channels or VGSC. And so the nervous system works very similar like it does for humans, in which you have openings and closings at those sodium channels that send messages to our muscles to help us move around, feed, do everything that we normally want to do. But when an individual is exposed to a pyrethroid, it keeps those channels or those pores wide open, and you have constant messaging to that nervous system. And eventually it causes muscle fatigue and death because those muscles are working so hard they never get a chance to rest or break. So pyrethroids work by contact or ingestion, and they would be considered broad spectrum. So they attack or are effective on a wide variety of insects. So I have some video here that shows the exposure over time to soybean aphid with pyrethroids. This is a healthy looking soybean aphid in a petri dish under a microscope. She's fairly active as she's walking around. She's not tapped into food, and so she's looking for food by walking. She's using her antennae to kind of sense the world around her, and she can get along pretty good when she wants to. The second individual has been exposed to pyrethroids, and it's been a few hours, and you can see that she is becoming uncoordinated. The rapid fire of the muscles are starting to take effect, and she is becoming uncoordinated and having a little bit trouble walking around. And this third video is an aphid that has been exposed about 24 hours ago to a pyrethroid. And she kind of looks like a tipped over cow. She is not able to stand and she's certainly not able to walk. She's not feeding anymore. The muscle twitching is becoming very fatiguing, and she is not going to live very long after that exposure. So that's how pyrethroids work. I wanted to compare something like a pyrethroid, which would be an older chemistry, to something that is more modern. And I'm going to use the example of a fetopyropen, or commonly known as safena, in which it also attacks the nerves and muscles, but it specifically attacks or changes the feeding behavior of fluid feeding pests. So it's a product that's great for aphids, psyllids, leafhoppers. And so the way that it works is, as an exposed individual, the active ingredient would target the mechanosensors in the joints of the legs and the feeding stylet. And ultimately, you can see the stylet here of these two aphids are not able to successfully pierce tissue and take up phloem anymore. And so it is a starvation tactic because eventually they're just going to run out of energy. And so I point this out because I've had a few people this year talk about safena in the field, and they say, I come back and it appears that aphids are still healthy, moving around, and they're not dying or they're not twitching like I would see for organophosphate or a pyrethroid application. But to know that it takes a little bit longer for this active ingredient to kill because it works through starvation. And as a newer chemistry, it is a bit more targeted and practically non toxic to honeybees. So for those farmers that maybe have hives around the farm, this is a great option, not only because it's effective on soybean aphid, but it is non-toxic to a lot of other insects like honeybees. You may be wondering how insects develop resistance, and that IRAC video does a nice job of giving the highlights, but generally, the most common type of resistance is a metabolic, in which the exposed individual can just detoxify the enzyme. There is also a target site resistance, in which it has modified binding. The target site resistance is most common in aphids, in which there's a mutation of that voltage gated sodium channel gene region. And so it's commonly described as knockdown resistance, or KDR, and super KDR for having multiple mutations or resistance in an individual. There is usually a consequence or a fitness cost for individuals that develop resistance, say, to an insecticide. So there's a wide body of literature that says resistance traits carry fitness costs, which could be described as smaller in size. Maybe they don't live as long, or it takes them longer to become an adult. They don't produce as many offspring. They lose some of their cold hardiness and they're not able to survive our cold Iowa winters, or they just have a reduction in alarm pheromones. So they don't respond to those environmental cues like temperature, reduced food quality, or the presence of predators. And the literature also suggests that the fitness costs are such an extreme consequence that the number of individuals that have resistance usually starts off very small in a population, and it develops at a very slow rate. And so that's the idea with limiting the exposures. We are reducing the rate or the number of individuals that develop resistance, and also the magnitude. So you can be a little bit resistant or you can be a lot resistant. And we want to limit or delay that resistance rate and magnitude as much as possible. I wanted to finish up by talking about some of the latest research updates that are happening at Iowa State. And the first one is back in 2016 is when I started to hear about pyrethroid failures, and some of the colleagues that I had in Minnesota also heard about them as well in commercial soybean. And so the specific example in Iowa was in a field in northwestern counties. I went and collected aphids from those problem fields, as well as my colleagues collected aphids from Minnesota. So what the applicator noticed was that a spray went on July 25th, and he sprayed a herbicide and an insecticide. Reveal would be a very common pyrethroid. He went back, thankfully, to just check and see how the spray did, and he noticed quite a few survivors and maybe even more aphids than before the first spray. And so he kind of blamed it on himself and an applicator issue. And so he went back in about 2 weeks later, and he went in with the same product again and sprayed. And he came back a few days later, and that's when he noticed a lot of survivors again. He got in contact with the field agronomists in that area, and they got in contact with me. And this process of collecting aphids from those problem fields is really the first step in confirming resistance. And so you can either do that through a leaf dip, in which you cut a disc out of a leaf and treat that leaf, or you can use a glass vial bioassay in which you coat the glass with different concentrations of an insecticide. But both ways, you are taking individuals from those problem fields and you're screening them on small batches. And so you put them on the leaf or put them in the vial and count the number of survivors after 48 hours. And we would compare the mortality ratio of aphids from problem fields from aphids in the lab that have never been exposed to an insecticide. And what we found from that 2016 collection is that the aphids in Minnesota and also Iowa had an average 39-fold reduction in susceptibility to lambda-cyhalothrin and bifenthrin. So those are two of the most commonly used pyrethroid active ingredients in soybean. A little bit later, we did a similar study, and it showed basically the same thing. About half of the aphids that we collected from problem fields had reduced susceptibility to pyrethroids. In practical terms, it meant that these aphids that were effectively sprayed with the fully labeled rate had the propensity to survive. And sometimes about half of those aphids survived, but a lot of times you had virtually no suppression in the population and a resurgence in aphids because they lacked competitors at that time. So they were thriving in an environment where they could have optimal reproduction. At the same time, we had some very smart entomologists, including Brad Coates, who is a USDA scientist at Ames Lab, decided to work on molecular markers to hopefully speed up that process because it is fairly time consuming and it does take a lot of people power to do those glass vial bioassays. And so he worked on collecting problem aphids from problem fields, and instead he could use large batches of dead aphids and screen them through a high throughput system. And over time, Brad and other entomologists were able to sequence 4 different KDR and super KDR mutations for pyrethroids for soybean aphid. And we had a graduate student who, with the help of the Iowa Soybean Association, continue or validate this work by collecting aphids for over 3 years. And he was able to get over 50 fields sampled. 10 of those were sampled with pyrethroids, and he was able to screen over 3,100 aphids using these molecular markers. And unfortunately, what he found is almost nearly every single aphid had at least one of those KDR or super KDR mutations. In fact, 42.5% had one mutation, 40.6 had two mutations, and just under 4% had three of those mutations. And in fact, 87% had one of those four known mutations that had been previously sequenced. Another 13%, or that blue wedge in the pie chart, had a KDR mutation that has not been sequenced yet. So, still more information to come on that. We only had 0.1% of the population considered totally susceptible to pyrethroids. And in other news, another graduate student did some further work, and he was able to demonstrate the heterozygote mutations were resistant as well. So that means an individual had one dominant and one recessive gene, but they still were able to express mutations and would survive a pyrethroid exposure. And kind of a worst case scenario, he was also not able to demonstrate any fitness costs observed with these KDR or super KDR mutations. So That means there's no consequence. They're not smaller, it doesn't shorten their lifespan, and doesn't reduce the number of offspring that they can produce. And remember, soybean aphids, if an individual is resistant to pyrethroids, immediately her daughters and granddaughters are resistant as well with no consequence. I asked Ashley Dean to summarize some pyrethroid performance data from our evaluations over a 10-year period to see if we ever had more aphids after a pyrethroid application. And so she estimated the percent difference when we sampled before a spray and then after a spray. And we compared that to our untreated controls. And you can see there's 3 years, 18, 21, and 24, where we actually had more aphids after the spray than before the spray. So ideally, what you like to see from an effective application is a reduction in aphids. You want the number to be below that zero. But in 3 years, we actually had more aphids. And I think that's what people were seeing if they were checking after the spray this year, is that they didn't see a decline and they actually saw more aphids than before the spray. Again, in the absence of competitors, and if you had ideal temperatures, you could have a resurgence very easily in a variety of growing situations. I just wanted to share a few other ongoing research related updates. The work for soybean aphid management continues with the concept of trying to reverse pyrethroid resistance. And so, with the help of Brad Coates, he is working on an RNAi based silencing probe. The I stands for interference, in which we can treat pyrethroid resistant soybean aphids with this probe and they become susceptible to pyrethroids again. It's pretty wild. I actually don't fully understand the process, but right now they are working on an individual aphid level. And what they're hoping to do is once they get that system worked out with those probes and exposures to those pyrethroid resistant individuals, we can scale up and we can start to work in the greenhouse and eventually get to a point at which we can spray small plots and maybe even commercial fields. So this is very early on research, but more details to come in the future. But it's very exciting to think about RNAi-based silencing tools. Now, the access to chlorpyrifos has been changing in the last couple years, and this is honestly stressful to people who are looking for alternatives to pyrethroids, knowing that pyrethroids largely aren't going to be effective anymore. One of the go-to products would be an organophosphate known as chlorpyrifos, but the tolerances have been revoked. They've been they're under exemption. And as far as I know, at the time of this recording in December 2024, you would be able to use chlorpyrifos in soybean and alfalfa in 2025. The tolerances are allowed. Now, since they were first revoked, some companies have just opted out of making chlorpyrifos. So the access may be limited if you're trying to use something else besides pyrethroids. So that is going to be something that I will know more about during the 2025 growing season and beyond. And just to let you know that I continue to do efficacy evaluations for new active ingredients, new formulations, trying to get away from pyrethroids as much as possible, and looking at targeted chemistries. And so I will always have updates every single growing season on recommendations for the future. So if I were to create a plan for you or for your clients that work in soybean or farm soybean, is to continue scouting this erratic pest. Most of the time, we don't have to worry about soybean aphid because they will not cause economic injury. But if we consistently scout and use the economic threshold, I think people will find they're breaking even on their control costs and hopefully making a profitable decision. Maybe 1 out of 4 years is based on my experience. When it is time to spray for aphids, use fully labeled rates. And again, we just want to avoid pyrethroids whenever possible because it can cause a resurgence and with the lack of competition from other pests. If you are using insecticides in soybean, it's important to think about alternative group numbers, especially if you happen to be spraying twice in one season. So I've listed some groups and some subgroups there as options. Again, the accessibility or the ability to actually buy and apply these products is something that is going to be a year by year consideration. If you happen to have aphids late in the season, it is a judgment call when you spray at R6. Also, pay attention to the pre harvest intervals later in the season. Some of those are up to 60 days and it may interfere with the harvest schedule. I really recommend using high volume and pressure. We want small droplets to be touching the aphids for our greatest chance of contact and exposure and hopefully knockdown of that population. And really, the only way to know whether a product is working or not is if you go back in after the reentry interval, count the number of aphids post spray. There should be a reduction. If not, you have to ask yourself about the application and also the performance of the product as well. And having untreated check strips is the most consistent way to say this strategy for scouting and treating or not treating was a profitable choice for you that year. So I want to thank you again. I have field notes that recaps the main points and provides some additional resources. I thank you for listening to my talk in Crops TV, and I hope you enjoy the other episodes.